Hello my people, welcome to SMASHFace Biology. I am Mr. G, and in today's episode, we are smashing translation. Translation is the process by which we decoded mRNA transcripts to make proteins. And so in today's episode, we will break down the genetic code, then we will break down the process of translation. We are doing this at first year university level, and so with that being said, it's time for us to smash. When we transcribed DNA, one type of molecule, into RNA, an entirely different type of molecule, that process still made sense because both of the molecules are nucleic acids. They share the same type of subunit, and so they can interact with each other in a way that made sense through base pairing. Scientists were puzzled by how we went from mRNA into proteins. Proteins are made up of amino acids, an entirely new type of molecule. And so this led to the development of what is known as the adapter theory. And what the adapter theory says is that basically there is a molecule that is an intermediate that helps us convert RNA into proteins. Check it.
mRNA transcripts are composed of a sequence of 4 nucleotides. Proteins are composed of 22 amino acids. It is impossible that for every nucleotide there'll be an amino acid, because that is basically 4 amino acids only. So scientists started thinking about making combinations of nucleotides. And so if we did combos of 2 nucleotides at a time to compose an amino acid, that number is still impossible because that it gives us mathematically only 16 options of amino acids. So we're still short. Scientists then thought, what if we do combos of 3 nucleotides at a time? Ding, 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 ding. That was the correct answer because there is 64 different possibilities, as seen in the genetic code table. The genetic code is sort of like a conversion table where each combination of 3 nucleotides are decoded into an amino acid. Each combination of 3 nucleotides are known as codons. To decode codons and make proteins, we undergo the process of translation. Translation has 3 major steps. The first step is initiation. The second step is elongation, but there's also a slight movement known as translocation that is also crucial for this step. And then finally, we have termination. Here's what we need to undergo translation. Well, first, we're gonna need our mRNA transcript. And so if we have a prokaryotic mRNA transcript, you can tell it is so from the lack of the 5' cap and poly A tail that you would see otherwise in eukaryotic mRNA transcripts. We would also need charged tRNAs. And what charged tRNAs means is that they have an amino acid at the acceptor stem. tRNA are non-translated RNA molecules that function as our adapter molecule. They contain a 3 nucleotide sequence known as the anticodon, which is complementary to our codon nucleotides. Through the help of the enzyme aminoacyl tRNA synthetase, amino acids are added onto the acceptor stem of tRNAs. We would also need ribosomes, which are pretty similar between prokaryotic and eukaryotic cells, except in that they have differences in size and composition of subunits. The large subunit has 3 sites for binding tRNA molecules. The A site binds aminoacyl tRNAs, tRNAs that are charged with amino acids. The P site binds peptidyl tRNAs, the tRNAs that are holding or binding the growing peptide chain. And the E site, known as the exit site, binds uncharged tRNAs. So once tRNAs deliver their amino acid and they are uncharged, they exit from the E site. Note the directionality of tRNA movement through the ribosome from A to E site. We also need the ribosome's small subunit. It binds the mRNA transcript, and this is where translation starts. Through initiation, mRNA is bound to the small subunit. Then a methionine tRNA is recruited into the start codon. This leads to the recruitment of the large subunit to bind with the small subunit. The next step is translocation, where actually the ribosome is moving through the mRNA transcript. The methionine start codon tRNA moves from the A site to the P site, and then the subsequent tRNA is recruited into the A site. The next step is elongation, where the peptide from the start codon methionine is passed onto the tRNA in the P site now. We translocate one more time and recruit the appropriate tRNA into the A site. Pass on the peptide chain onto the upcoming tRNA molecule and continue sliding our tRNAs and translocating. Note that the ribosome reads only 3 nucleotides at a time, or a codon, and that the tRNA being recruited into the A site has the complementary sequence to that being highlighted from the transcript. The polypeptide chain continues to grow until we reach what is known as a stop codon. Stop codon signals the beginning of termination, where a release factor is recruited onto the A site, and that leads to the release of the peptide chain. This peptide chain, or amino acid sequence, needs to be folded to become functional proteins. And that concludes translation. Best of luck to you on your studies. Remember to always